All right, I think we'll go ahead and get started this morning. It's really an honor and a privilege to introduce Giedre Krenciute as our speaker today. Giedre is a associate member at the Center for our Department of Bone Marrow Transplantation and Cellular Therapy at St. Jude Children's Hospital. So Giedre received her bachelor's degree at Vilnius University in Lithuania before moving to Texas and completing her PhD at the Baylor College of Medicine in biochemistry and molecular biology. She then went on to do her postdoctoral training with Malcolm Brenner and Steve Gottschalk's group, also at Baylor, in the development of new and innovative cellular therapies focused primarily on CAR T cell therapies. Since starting her own lab at St. Jude, she's continued to focus on developing innovative CAR T therapies for pediatric brain tumors, where she's focused on establishing next generation CAR T therapies to target pediatric brain tumors, enhancing their persistence and efficacy using different genetic manipulations, understanding mechanisms for tumor escape, and understanding CAR T-cell memory programs that control their long-term effector function, and defining how these immune cells interact with the tumor microenvironment using immune-competent brain tumor models and others. Giedre's been very successful in terms of funding and publishing high-impact work, and importantly, the discoveries that have come out of her lab have supported clinical translation, including a clinical trial on the local regional delivery of B7-H3 specific chimeric antigen receptor T cells for pediatric patients with primary brain tumors, which opened up in the spring of last year and is actively recruiting. So I'd like to just say thanks for coming, and very excited to hear about your work today. Thanks.
Thank you both. Thank you so much for your kind introduction and invitation. Thank you all for coming. I know it's early. So as Bo said, and the rationale for why we need better therapies is pretty straightforward. So far, we know that outcomes are pretty poor, and one of the major obstacles is very long-term toxicities. And immunotherapy, especially T-cell-based immunotherapy, has potential to address this lack of better treatments, because immunotherapy is targeted, especially if you use CAR T cells. It does not rely on cytotoxic mechanism of conventional therapies and can reactivate the immune system. Now, I know you all might be familiar with the CAR T cells, but I'm just going to briefly go through them to make sure everyone is on the same page. So CAR T cells are those T cells engineered with this chimeric antigen receptor, a highly synthetic molecule that consists of 3 functional domains. That is antigen recognition domain, that is in charge of tumor cell recognition, co-stim domain, that is in charge of T cell proliferation, and CD3 zeta domain, which is causes T cell activation. And so altogether, again, it's transducing T cells and make T cells then tumor cell specific and redirect those T cells to the tumors. So the goal of my research program is to really answer the question that was the title of my talk, is what will it take to make CAR T cell specific for pediatric brain tumors? And really, we do this by by working on 3 big projects. One is improving CAR T cell efficacy by genetic engineering approaches, then trying to figure out how we can synergize CAR T cell effector function with tumor microenvironment, and finally, looking into possibilities of combination therapies if it's clear that one modality probably will not make it work. And then all these projects are have high clinical translational aspect. So everything that we do is towards clinic, and then we do clinical trials, and then that informs what we're going to do in the lab. So we have this circle going. At the moment, I focus in the lab, we focus on DPGs, highly aggressive pediatric brain tumors, medulloblastomas, and ependymomas. So when I first started my lab at St. Jude, the question was, what is the antigen that we should target for pediatric brain tumors? It was not, for adult tumors, we had a lot of information. And at that point, there's 5 well-established targets for adult tumors. So that was B7-H3, GD2, IL-13 receptor alpha 2, EphA2, and HER2. So what we decided to do is to take those well-described antigens for adult tumors and scan pediatric brain tumors and see which one is the best for establishing CAR T cell therapies. And so here's this heat map is a summary of everything we did. So we used commercially available pediatric cell lines, and then about more than 50 patient-derived xenograft cell lines. And we used flow cytometry to look at those 5 antigens. So a couple lessons that we learned, that all cell lines, commercially available cell lines shown in here, they all highly express all the antigens, indicating, they are not really representative of this heterogeneity of the antigens that we see in patient samples. And interestingly, HLA we use as a control to really discriminate between human cells and murine cells, because those were propagated in mice. And what we saw that in medulloblastoma, we see a downregulation of HLA, again supporting that CAR T cells should be probably considered for these particular tumors. And then the final message was that B7-H3 is the antigen that is highly expressed throughout multiple pediatric brain tumors. So that justified us to really make B7-H3 specific CAR T cells. I'm not going to go through all the details, but we did make B7-H3 specific CAR T cells. We showed that those CAR T cells are effective in preclinical models, and those are safe when we used immune competent mouse models. And tons of all that data led into opening this clinical trial for pediatric brain tumors. And we have 2 cohorts. First is for all refractory relapsed pediatric brain tumors, and the second cohort is the APGs. And we do require H score for B7-H3 that is higher than 100, unless it's cohort B, which is the APGs, which we don't usually biopsy them. We see in this particular trial, we have total of 6 infusions, 3 infusions at the beginning, a week break, and then followed by another 3 infusions. And the CAR that we used, it was for B7-H3 specific CAR T cells with CD28, CD3 zeta signaling domain, and we also did additional engineering, adding 4-1BB ligand to give extra boost to these CAR T cells. And as Chris Renzo in our department published in 2020, and we showed in brain tumor models that this molecule, this particular setup really works the best. So we decided to proceed in clinic. The first aim of this clinical trial is to really evaluate the safety and efficacy of these CAR T cells. And then the second aim is all correlative studies. Really, we collect CSF, blood, CAR product, and we look at all kind of correlatives to answer and study T cell biology, CAR T cell biology, treatment response correlatives, metabolic profiles, and then impact of immune cells. And then we put that together and then proceed with reverse translation to inform the next arm of our clinical trial. So again, as Bo mentioned, it opened last year in April, and enrollment was quite brisk. We have phone ringings every week, people wanting to enroll in this trial. So far, we screened 25 patients. We evaluated B7-H3 expression in 21 tumors, and you can see the age of participants are indicated here. So really wide range of participants. And B7-H3 expression, again, is we only had 4 that did not qualify to the trial because of the low B7-H3 expression. And then here's the distribution between different pediatric brain tumor disease. And the ones in dark circles are the ones that were treated in clinical trial. So again, total since, and this is just the update as of August, we actually treated additional 2 more patients, and 2 more patients are in progress of product being made. Total, we had 43 infusions. And because of the time limitations, I'm really not going to go into details on what we see so far, because I want to just talk about what's the next for this. But same as all the other 4 clinical studies that are published so far, we also see that those CAR T cells are safe. However, we do see partial or a non-durable response. So really the next question is for us, what's next? And what's next is how we can address partial T cell response. And now I would like to share the 2 stories of what we're working on. So one, overcoming limited T cell persistence through gene editing, more specifically knocking out DNMT3A and other negative T cell regulators. And then the second part will be on understanding brain tumor microenvironment and the effect on the CAR T cell efficacy. So proceeding with the first topic, it is well established that when we have CAR T cells, those are exposed to tumor cells. CAR T cells kill tumor cells and they expand. However, upon multiple exposures to tumor cells, CAR T cells eventually fail and start contracting, and they basically stop working. This phenomenon is better defined as exhaustion, which is further defined as lack of proliferation, lack of killing, lack of T cell persistence. And the mechanism is really not clear at this moment, but there's a lot of labs studying it. And one approach to do that, and Bo is also doing similar things, is to use CRISPR-edited CAR T cells to improve them. And the idea is that we use CRISPR approach to knock out one of the negative regulators, and then we will have those better CAR T cells that now we can expose to tumor cells multiple times, and they will expand and persist. And when tumor is no longer there, only then they will contract and persist till, let's say, tumor reoccur. The question is like which negative T cell regulator to knock out, and there's multiple screens coming up, different modifications that you can, or different favorite molecule that you can pick. What we decided to do is to knock out DNMT3A because it was shown actually by Ben Youngblood that in chronic viral infection model that DNMT3A controls exhaustion genes. And when you knock out DNMT3A, you have a promoter open for those DNMT target genes that lead to naive-like T cells. And those T cells have this sustained effector function and better persistence. So really we just asked, and again, everything was done in chronic viral infection model. So we ask a very simple question: can blocking of epigenetic programs improve CAR T cell function? And really, this was a teamwork in collaboration with Ben and Steve Gottschalk, with first and leading author Brooke and Caitlin. So what we did, we knocked out DNMT3 CAR T cells, and here is just showing how we do that in the lab. And you can see that in multiple CAR T cell, multiple donors, we can get very efficient DNMT3 knockout, and that knockout does not affect CAR transduction. So we have the same product as control edited CAR T cells. Then we proceed with this, what we think is a key assay. So in this assay, we take our CAR T cells exposed to tumor cells at 1:2 ratio and let them sit for 7 days. At that point, we then count CAR T cells and re-expose to tumor cells. And we continue with what we call this repeat stimulation assay until T cells stop expanding or stop killing. And here's just illustration for HER2 specific CAR T cells against U373s. And here you can see that upon first three stimulations, there is no difference between control and DNMT3 knockout CAR T cells. However, at the later stimulations, you see that control CAR T cells start crushing down, where DNMT3 continue to expand. So that's great. So we see better expansion and better persistence. At this point, we also ask, well, now that we have this great expansion, is it actually specific to tumor cells, or maybe we formed another malignancy? So what we did, we removed tumor cells and then checked 7 days later using flow cytometry. And what we found that really when there is no antigen, T cells stop expanding, indicating that this expansion and persistence is antigen dependent. We also ask, well, is the cytolytic activity still working at the first stimulation? So here you can see that in control and DNMT3 knockout CAR T cells at the first stimulation, they kill exactly the same. However, the later stimulation control of CAR T cells start losing their cytolytic potential, where DNMT3 CAR T cells still can continuously kill, indicating that DNMT3 again has sustained effector function. So then what we did, we really wanted to look at the extrinsic factors that might drive this improved persistence and expansion. So here, what we did is after first co-culture, first stimulation, 24 hours, we collected cytokines and look at different cytokines using multiplex assay. And we saw different changes in different cytokines, but what we consistently saw is IL-10 being upregulated. We did the same assay at the fourth stimulation. Again, after fourth stimulation, 24 hours, we collected supernatant and run multiplex assay. And once again, we see IL-10 being consistently and highly upregulated. Well, that was interesting. You know, it's not what we expect to see because IL-10 is known to be a suppressive cytokine. So then we ask, well, is that actually needed, or this is just a consequence of this, I don't know, the stimulation of T cells, and actually it's not driving T cell persistence. So what we did here, we knocked out, now we have control, DNMT knockout, IL-10 knockout, and double knockout of DNMT3A and IL-10. And this is in green, and you can see that CAR T cells lose their potential to expand, and really this is a log scale, so it's not seen, but you can see that it looks exactly as a control, indicating that IL-10 is really needed for T cell expansion. Then we did exactly the same assay, but we add back IL-10, and now you see that the double KK knockout was DNMT3 and IL-10, and when you add IL-10, now it looks exactly as DNMT3 knockout, once again indicating that IL-10 is beneficial for this. And this is also recently, you might be aware, but this paper just came out showing that IL-10 expressing CAR T cells actually do better. So that's just supports our findings. And we are looking into mechanism of why IL-10 is helping here, because again, it's not what we expected because it's suppressive cytokine. The only one thing that I can tell, it's not the conventional signaling IL-10 pathway through STAT3. We don't see any changes in that. So because we were looking at the because we were knocking out DNA methylation protein. So then we looked at that DNA methylation programs using a whole genome bisulfate sequencing. And here what we did, we collected cells after force stimulation, sorted for CD8, did DNA extraction, performed whole genome bisulfate sequencing. And that was done in collaboration with Ben, Caitlin, and Yiping Fan to look at changes that we see after DNMT knockout. There's a lot of course, changes as you lose this protein, but overall what we see is that DNMT knockout CAR T cells retain naive-like T cell programs. There's a lot of unmethylated promoters and genes involved in prevention of terminal differentiation and functional memory development. And just to give you one example, genes such as TCF7 that is known to regulate T cell self-renewal and differentiation. What Ben showed previously, if you take healthy donor and sort naive cells, TSCMs and TMs, you can see very particular methylation profiles. So when we looked at the two different CARs with control and DNMT knockout and look at the promoter of TCF7, you can see that the promoter in the knockout really resembles TSCM T cell phenotype. And then we confirmed that by flow cytometry looking at TCF7 at the protein level, and we indeed see that TCF7 was upregulated in these conditions. So then we proceeded with in vivo experiment. We did multiple CARs, multiple different in vivo models, but here I'm going to show you only the gold standard CD19 CAR. So here's the setup of the experiment. We used BV173 tumor cells, and then did systemic injection of 2 million CD19 CAR T cells, control or DNMT edited. And really response was great. We see much better anti-tumor control. And we see actually two complete responders and long-term survivors, and that led into significant overall survival. Now that because we had these two complete responders, what we decided is to re-challenge them at day 113. So we re-injected additional dose of tumor cells. And what we saw is a continuous protection from the tumor formation. But at that point, it's 113 days. Who knows what's happening here? It might be that just, mouse is having all the human T cell, or just tumor is getting rejected. So to really overrule this possibility, what we did at 148 days, we implanted now BV173 tumor cells where we knocked out CD19, and those tumor cells implanted. So what we see in here, it's really improved persistence and tumor control. It was highly specific for CD19 positive tumors. So to summarize the work that we've been done at that point, or everything that we observe is DNMT knockout, is that a deletion of DNMT knockout improves CAR T cell expansion as well as persistence, and you have sustained effector function throughout multiple stimulations. We also show that DNA methylation patterns are consistent with less differentiated T cells, which explains their performance and persistence, that IL-10 is important and it promotes CAR T cell survival. And then that I didn't talk about it, but we also formed DNMT3 target gene list. And then we looked at these genes in the clinical trials, and we found that there's correlation in CAR T cell efficacy in the clinic when you look at the gene expression of DNMT3 target genes. And what else we did is that we then confirm all the key findings in multiple specific CAR T cells, multiple tumor models, and we always continue seeing the same improvement in DNMT CAR T cells. So we published late in 2021. The next year, another paper came out showing that the 2 is maybe better. This is another genetic modifications. And really, so the question is like, which one is now the best for the brain tumors? Every single group, including ours, we publish one genetic modification. It's always good, but we never really compare them. And so a brave postdoc in my lab, Megan, she decided to take on this project to really compare side by side two well-established epigenetic modifications in pediatric brain tumors, more specifically medulloblastoma. So she studies group 3 medulloblastoma. She showed that B7-H3 is highly expressed in all cell lines that we use. She established also growth curves to show like these tumors are highly aggressive, kill animals within 20 to 30 days. And she used these models to study now epigenetic modifications. So she used B7-H3 specific CAR T cells, again, the ones that we used in multiple, in the clinical trial, just without 4-1BB ligand. Transduction is great, even if with the gene knockouts, and you see that when we knock out TET2 or DNMT3 at the baseline, there's no difference. So, you know, I know it's a lot of graphs, but you just see there's no difference. So that's great. At the baseline, we have exactly the same product. So the next slide I'm going to show is going to be a lot of data. So so we do the same repeat simulation assay. Again, we take medulloblastoma cell lines, we took multiple ones, and we exposed to TET2 and DNMT3 edited CAR T cells at multiple times for 7 days and just keep going and going. So this is what we have. Let me just walk you through this. So what we have is 3 different medulloblastoma cell line that express B7-H3 at low levels, high, medium levels, and high. Then we have B7-H3 edited CAR T cells with control, TET2, and DNMT3A. And then we have 6 donors, 6 CAR T cell donors that we tested this because, and I'm presenting this data separately because to those who are working with CAR T cells, you know the headache of donor to donor variability, just to show how variable it is overall. But so lessons that we learned here, if you follow this purple or pinkish line, which is DNMT3 knockout, you can clearly see that in every single condition, DNMT3 knockout consistently is the best, except a couple of the occasions, a couple of donors where TET2 is actually better, which now is really in line with Michelle Sadelain published that TET2 knockout works better in donor-dependent manner. So there are different drivers that make TET2 better. But I also want you to see, and you know, that we don't really see dependency on how much antigen we have on a cell surface, right? We have low, medium, and high, and you know, expression here, T cell expansion is here, okay. This is amazing expansion, and yet the cell line that has high B7-H3 expression, we don't see much expansion. Once again, showing that donor variability, but nevertheless, DNMT3 consistently show much better CAR T cell expansion. So that was great. And again, tons of work was done. I'm just going to summarize that there is really DNMT3 knockout and TET2 knockout CAR T cells show very different profile. And so in DNMT3 knockout, we see enrichment of CD8s. We see again IL-10 being upregulated always. Cytotoxicity is continuously to be better compared to TET2. It actually starts going down after multiple simulations, and TET2 response is donor dependent, where DNMT we always see improved. And then antigen density, we don't really see any dependency on that. So once again, we proceeded with in vivo testing using the same 3 cell lines that has different B7-H3 expression levels. So here's a setup. We implant our tumor cells, different numbers depending on the model that we use. And 2 weeks later, we inject our CAR T cells directly into tumor. And that is usually 2 million bulk CAR T cells that were DNMT3, TET2, or control edited. And here's the data. Again, I'm showing all at the same time, and you can easily see that there's no difference. Nothing that we saw in vitro. So that was quite disappointing. So CAR T cells work a little bit better, but the TET2 or DNMT3 knockout is not really working well. So we're still proceeding. So now the question is like why it's not working, trying to understand. One idea in here is that these tumors are highly aggressive, and we just don't give enough time for T cells to establish this better persistence. So you need a slower growing model, and that's why we always, so maybe the DNMT knockout would be better in something like CD19, where we see late relapses where T cells can persist. But in these highly aggressive, fast growing models, we need something better than DNMT. So we do have a couple experiments in mind that we want to do in vivo and see if we can actually see a difference. But at this point, we just see a marginal increase in nothing like we saw in vitro. So to summarize this part, we believe that gene editing is really a powerful technique. We can make improved CAR T cells, provide new knowledge about CAR T cell biology, and then inform next generation CAR T cell design for the clinic. And then ablation of different negative T cell regulators improve CAR T cell effector function. So we see enhanced CAR T cell cytotoxicity, expansion, and persistence, and better function in vivo. Yet the question which one is the best is still not clear. Now Megan, what she decided to do is to test more epigenetic regulators. After the TET2 story came out, then another gene pop-up was SUV39H from Sadelain's group, as well as KDM4A from other groups. So really, we're now knocking out all of them and trying to see what's going to happen. We also, Hombo Chi at St. Jude also showed that Reganase 1 knockout is great. And again, Megan and my senior scientist in the lab, Jorge, they are testing everything in pediatric brain tumor setting. Just a little bit more thoughts of which one is the best. we're trying to put together a review of all the known negative T cell regulators, just the genes that you can knock out. And so far to date, there are about 82 published negative T cell regulators. So which one do you pick? And go to your chair or whoever is in charge of which next clinical trial we're going to run, which one do you pick to say which one the best? And we really struggle with that to justify the generation of next arm of clinical trials because it's expensive, lengthy, and we want it to work. And the way we start thinking of this is really that we have to organize them in functional buckets. So there are CAR T cells that, maybe they need to be a better killers. So if that's the case, we maybe need CD28's costim domain, Braganase 1 knockout, RAS-A2 knockout is another one, or this PDZ domain that we just recently published and showed that immune synapse will make them better killers. For persistence, maybe the 2, DM3A, and maybe in that case, or C4-1B costim domain. And if you need extra activation because it's not, there's a low antigen expression level, maybe you can express cytokines, target MyD8 signaling pathway, and a transcription factor role. However, whichever one would pick, they also have to consider disease. Is it aggressive? Is this relapse? For example, if it's relapse, maybe then we do need better persistence because eventually we're going to clear the tumor, but they're going to relapse and we need floating CAR T cells in there. T cell donor, is it good or bad? We need to figure out the markers for that. And also which CAR T cell target it is. So there are some CAR T cell targets that are expressed really highly, and some of them are lower and are not able to induce potent CAR T cell signaling. So this is what we think. And then, how that's going to look in a clinical trial, we just don't know yet. But I think learning now more about tumor cells that we're trying to target and eradicate is the way to go to really inform this next genetic modification. All right, so moving to the next part right now, understanding brain tumor microenvironment. So here I'm going to present work done by my previous former postdoc in my lab, Dalia Haidar, who's now assistant professor at Children's National. And she studied how tumor microenvironment affect CAR T cell function. And this work has been published last year. So if you're interested, you can definitely read. And I'm just going to walk through the key findings that I believe are interesting. So previously, also, as we were preparing for the clinical trial, we needed to show the safety of B7-H3 specific CAR T cells. So we generated murine B7-H3 CAR T cells and showed that these CAR T cells are effective and safe in immune competent mouse models. And here we tested NGL 261 animal model and showed that you see quite potent anti-tumor response that leads in a good overall survival. However, not animal respond. At that point, we took the tumors out, checked for B7-H3 expression on those tumors, and they all are still expressing, indicating that it's not antigen escape. It's probably something related to persistence, or maybe because we used immune competent mouse models, it might be because of the suppressive microenvironment. And at that point, also, we only tested one costim domain in mouse setting. And we know from human studies that it's not enough to just say like, well, we take this costim domain and that's going to be enough. So we ask a simple question again, can changes in CAR signaling domain make them better? And also maybe one of them will function better in tumor microenvironment. To address that, Dalia generated an array of different CARs, really changing all kind of things in here. So this is our control CAR. So still recognizes B7-H3, but doesn't have a signaling domain, so it's not functional. And then here's our original CAR that has CD28 costim domain and mutated zeta. So we made a non-mutated zeta, we changed costim domain, we changed transmembrane domain, and then we created 4-1BB ligand that we tested in clinic. And right out of that, we had to exclude this one as it was not expressed on a cell surface. So we just decided not to study it. At the killing level, they all kill really well at a high effector to target ratio. But when you challenge them with the lower numbers, you see that you start seeing some differences. But overall, they all kill. Okay. Then again, we proceeded with our in vitro assay, repeat stimulation assay, same as in humans. Multiple rounds of stimulation. In murine setting, we do that for 3 days, no extra cytokine added. And what we're seeing here that all of them, except this one was 4-1BB costim domain, is not expanding really well. But the one with 4-1BB ligand in trans, the one that has additional costim signaling is really the winner here. It persists after 16 consecutive simulations, expands really well. Again, looked like a winner to us. And also what was great, because that's what we're using in clinical trial. That was also supported by different cytokines. So at the first simulation, we saw a lot of cytokines and it sustained, so interferon gamma, IL-2, but really GM-CSF, especially for the CAR that had 4-1BB ligand. So that was all great. So then we, and you know, like these studies are done so that we can eliminate some of them because it's really a lot and no one wants to do all of those in vivo. But then we thought like, why not to just like test all of them? Because maybe things will change. And then we can also additionally confirm that the ones that are not working in vitro will not work in vivo. So we did our in vivo assay. So here we again have GL261 mirroring GBM cell lines. We injected intracranially. 7 days later, we injected multiple murine CAR T cells. And then we followed tumor growth by MRI. We removed GFPFF loop that we used to do back in the days because of the possible immunogenicity. And so this is the data. So here we have control CAR, not really working. This is our CAR, original CAR that works pretty well and still working great. And here's all the other CARs. So really, none of these modifications were able to improve our CAR. And so to just kind of make a little things a little simpler, so I decided to kind of summarize in this table. So what we see, we have here all these different CARs that we made. This is our prototype CAR, the best one. And then here's in vitro function. And we see that, you know, some of them are better, some of them are worse, but they all are functional. They all expand, they all kill. Yet in vivo, though, we have a very different response. Our prototype CAR works the best. Our CAR that worked really well also was for 1BB costim domain, did not work in vivo at all. Our winner CAR that worked amazingly well in vitro also did not work well. So, and then some of them that didn't work like well, worked also better in vivo. So really the question, what is happening here? Why? And you know, the difference that we have in vitro and in vivo is that we have a dish and then we have immune microenvironment. So really to test if this discrepancy between in vitro and in vivo science, findings are because of the tumor microenvironment, we decided to do a single cell RNA-seq and just to see broad changes what we see after CAR T cell injection. For that, we set up this in vivo assay where again we inject GL261s, allow GL261s to grow pretty big, then inject CAR T cells, and then 4 days later we take everything out. The idea was not to treat tumors, but really activate T cells and capture them at this peak expansion or at the peak time where they hopefully are functioning well. And for that, we decided to use 4 groups. So here we have control CAR T cells, we have our best working CAR, and 2 CARs, mediocre CARs, that show very similar overall survival right here. And the idea was like, well, if they show similar overall survival, maybe their environment will be the same. And then we can say like, oh, we can predict CAR T cell response. So here's just a big overview. We don't see any differences in frequency in big immune cell types. So we have lymphoid here, myeloid, and tumor cells, and you see that when you compare between treatment groups, there is not much difference. If you look at the particular markers for different immune cells, you can readily identify macrophages, T cells, microglia. And then because of that, now we can kind of go into particular sets. And because people are interested in seeing T cell response and macrophages, then we kind of just focus on these 2. So I'm going to talk about T cells first. So for T cells, we have 3 clusters that were all T cells. And again, when you look again and compare them between treatment group, again, not much differences that we can say like, well, that's maybe means something. But what we do see in these different clusters, which we have CD8 T cell cluster, CD4s, and then activated, we do that in CD8s, there are a lot of exhausted phenotypes popping up, and then in CD4s, we don't see that. We saw metabolic enzymes being upregulated, but then in activated T cells, which we predict to be CAR T cells maybe, they are all highly proliferating, they're all cytolytic. And then again, when we just kind of assume that maybe this CAR T cells. Now, there's no way for us to kind of label for CAR T cells, but we can do, because we know the sequence and it's very unique because of the costim domains and because it's a synthetic molecule, we can go back to RNA-seq results and look for that sequence. And that's why what bioinformaticians done, and they found indeed that in these 3 clusters, we can see T cells, CAR T cells. So we can detect CAR T cell mRNA. Interestingly, we also see CAR mRNA in macrophage settings. So that was interesting. So, and I'll get back in a little bit. So that's T cell observation. So really nothing that can tell us why some of them fail, because everything looked the same. So then we decided to look at tumor-associated macrophages. So we identified 4 transcriptional clusters. Again, if you look in the frequency, nothing that is screaming a huge difference here. But then we decided to look at the particular molecules that are associated with different macrophage phenotypes, such as immunosuppression, invasion, recruitment, M1s and M2s. People like to see those and see maybe we can distinguish and say like there's different macrophages present in here. So again, here we have control, which is not working CAR, best working CAR, and then 2 mediocre CARs. And again, as much as I would like to say we see like M2s or something like that, we don't see. We see all of them, right? We see subsets of genes that are linked to M2 phenotype upregulated, but there are subsets that are downregulated. Same for M1s. We see some of them being upregulated in control and some of them in the best working CAR. So indicating that maybe we need a balance for them to work, and we need different phenotype macrophages. One thing that we notice is this inverse pattern. So whatever gets, let's say, upregulated in control, we see downregulated in our best working CAR, or whatever is upregulated in our best working CAR is downregulated in control. So there are some markers. It's just like we just can't find a pattern. So then we decided to take those 4 transcription clusters and recluster them again to look into more granular details. That led us to identify identification on 10 additional macrophage clusters. And I like to present in a UMAP form in here. Once again, we have best working CAR, mediocre CARs, and control CARs. And you can see like right away that there's these 2 clusters, cluster 7 and 2, that are present in the best working CAR and not present in a worst working CAR or control. So that was interesting because they are present in the best working CAR. I decided to call them good TAMs. So now what are these good TAMs? Looking at when you compare the expression level, cluster 2 versus all cluster 7, again, we see this balance of genes upregulated M2 and M1 macrophages. For example, when you compare cluster 7 to all the other clusters, you see arginase 1, which is marker for M2s, THPS1, which is marker for M1s. Again, this really, we can't say that we see one or the other, but maybe rather a balance, and maybe indicating that maybe we need both of them in the response to CAR T cells. Because we found some CAR mRNA in macrophages, then we ask, well, can we find CD3 or T cell mRNA in macrophage clusters? And yes, indeed we can. We actually see a particular macrophage clusters having T cell mRNA. So that was interesting. And also it's interesting that only specific macrophage subclusters are show presence of CD3 mRNA, but not the good TAMs, which is cluster 7 and 2. So then again, we ask, what about CAR T cells? What about CAR T cell mRNA? And again, we see that we see enrichment of CAR mRNA in macrophage clusters, but not in a control. And that was not unusual observation. Christelle Makel just recently published a paper in Nature. She also showed that in clinical samples, you see CAR mRNA in macrophages. And her thinking was that actually, they showed that when CAR T cells get activated, they downregulate don't eat me signal CD47, and macrophages are eating them. So she generated phagocytosis resistant CAR T cells, but they also what's known in literature that when CAR T cells, but also macrophages are eating dying cells, and we know that CAR T cells can have activation induced death, so probably macrophages are eating those. The question is like, is it good or bad? Just because we see more CAR mRNA in this macrophages compared to control, we believe it's good. And the question is like, should we engineer, or how we should adjust? Our treatment is still not clear, but we're working on it. So, because we have this good and bad macrophages, the next logical step would be, well, let's just deplete those good TAMs. Unfortunately, we cannot find a specific reagent that can deplete macrophages. So then what we decided to do to just do complete depletion of macrophages using this molecule called BLZ 945. We pretreated animals, implanted brain tumors in them, and then looked at their and how CAR T cells respond. And I'm not showing in here, but this particular molecule does deplete macrophages very efficiently. So again, we injected CAR T cells, and here's the result, showing that once we deplete macrophages and inject CAR T cells don't work anymore. So it's right here, this dotted red line. And if we treat with vehicle and CAR T cells, and we still have macrophages present, only then we see good anti-tumor response. indicated that maybe we need some of the macrophages. Maybe macrophages are needed to some extent in eradicating tumors. So to summarize the second story, what I showed you, that tumor microenvironment is highly dynamic, and that tumor microenvironment composition is highly dependent on CAR signaling domain. And I think to me it was really mind-opening, because as simple as co-stim domain, which leads to a different CAR T cell signaling, will invite different immune cells and then will cause a different response of microenvironment, and then it may or may not, shut down the way CAR T cells are functioning. And I always try to jokingly say, like, are we over engineering CAR T cells? Because good CAR T cell then will cause this massive inflammation, and maybe we have and then shut down of the whole system. And then we also show that CAR T cell treatment can dramatically alter macrophage subpopulations. And then seems like there is a specific subset of TAMs, I like to call them good TAMs, that are needed for good CAR T cell response. Again, we try to figure out if there's any cell surface markers of those TAMs that we can maybe deplete somehow, and then go from there. And then here's the different boxes of how we see CAR T cells working. So if we observe complete response, we usually expect to see still T cells present. Those are still functional in the presence of good macrophages. And then if treatment fails, there's a lack of T cells, and we don't see good tumor-associated macrophages. And now that just to end was the question that I started: what will it take to make CAR T cells effective for pediatric brain tumors? So I believe it will take to have a good and safe pediatric brain tumor target, such as B7-H3, the one that we're studying, and others. Long persistent CAR T cells with sustained effector function. We can achieve that by genetically modifying CAR T cells with a knockout of different molecules. Here I only discuss genetic deletion of the molecules, but there's all the other array of different modifications that can be tested. And then really, the next big question is how to synergize CAR T cell response with tumor microenvironment. And for that, we really need to look at the longitudinal studies of the brain tumor microenvironment during the treatment to really see what is happening. While we did 4 days after treatment, what if at that 4-day treatment, our best working CAR was performing better 2 days before, and we would have gotten better response. So really, those complicated longitudinal studies are highly needed. Then careful engineering of CAR T cells. Once again, are we over engineering CAR T cells? And then selective, seems like we need a selective depletion of inhibitory molecules. Once again, this approach of like wipe it out everything, it doesn't seem like it's working. And with that, I would like to acknowledge all the people that were directly or indirectly involved in all the research that I presented right now. And this is my lab, my current lab setup, and then all the other people from St. Jude, especially all the brain tumor experts. Really grateful to have them on my team. And once again, thank you for your attention. Happy to answer any questions.
I have microphones for anyone with a question in the room.
Morning, great talk.
Good morning.
I had a quick question on your serial killing / serial stimulation assays. What was the decision with the addition of IL-15? And then during the outgrowth, did you see enrichment, or did you check for enrichment of your engineered cell population?
So I'm just gonna answer from the back. Yes, we did see enrichment for the engineered T cells. Well, we do NGS, so we always see knockout, but also we do see enrichment for CAR T cells. And also we see enrichment for CD8s in that. So IL-15, it's a great question, right? We always get, so like, of course, with IL-15, you're gonna get this. But if we don't add IL-15, some tumor cell lines, T cells don't expand enough. There's huge variability. And also we can get to the forced stimulation, let's say, and get control and DNA at the same time. And then finally, we can get them to push the persistence. We don't see like they crash so fast. So addition of IL-15 allowed us to push that to this like long-term persistence. And that's where we start seeing differences. And you can see that in some tumor cell lines, we needed to go up to 8 stimulation to start seeing DNMT3A, and sometimes the 2 start separating. And then, we made this, and there's this huge debate in our department, when and when not to use IL-15. And we decided for consistency, we always gonna use IL-15, again, to push them to really mimic persistence sedation. Now we, comparing side by side, 3.5 days co-cultures was done without the IL-15 and see if we can capture those sooner, because, we can see that some co-cultures go to up to 10, 12 weeks. And, you can imagine people getting upset when it gets contaminated like 10 weeks or so. And also we also still wondering, what IL-15 is doing. So it's a great question. We still stand by what we did, because we really wouldn't capture all these differences.
Any other questions in the room? Let me check the computer.
And have you thought of knocking out both the TET2 and the DNMT3A at a time to see how the cells are behaving, something like that?
Yes, so we did that. Megan did that when she did DNMT3 knockout, TET2 knockout, DNMT3 and TET2 CUV and all that. She did in vivo. There was in that particular donor, TET2 worked well, and then double knockout TET2 worked well. Then, so that's what we saw. We didn't do any in vitro testing because we were like, just this is enough, and we did in vivo, and that's what we saw. But again, TET2 is highly donor dependent in that setting. Probably that's what led to this, and that's what we saw. Do we need to knock out both of them? I don't think so. I rather would go with something that is in different pathways. So maybe, I'm biased, but we also study RAS-A2, and RAS-A2 makes CAR T cells better killers. So I wonder if we can knock out RAS-A2 to kill T cells, tumor cells faster. And then later, when tumors start reoccurring, maybe we need DNMT3A to start wiping out the remaining tumors. We did that experiment, it didn't work anyway. So. But I think there's really model dependent.
Hey, nice talk.
Thank you.
So I'm curious about the route of administration. So, some of the tumors types that you're interested in treating show leptomeningeal spread, like you see in medulloblastoma.
Yeah.
And so do you think that another type of assay has to do with whether the T cells will the CAR T cells will be functional if you deliver them right to the cerebrospinal fluid and assess their ability to migrate and find tumor, or are all your assays in mice direct intratumoral injection?
So right now we're doing directly intratumoral. In clinic, we do intraventricular. In mouse, we decided not to do it because that would lead to 2 spots for drilling. So we wanted to avoid that. We know that T cells can migrate. Back in the days when we used to do systemic injections, and we would get tumor clearance, and then 2 clinical trials are showing that when they did systemic, so that one's one from Marcella mouse and the other one from Baylor, showing that when you inject systemically, they do travel to the brain. I don't know if anyone looked at that. Oh, Christine Brown showed if they do intracranial injections of CAR T cells, there was metastasis in the spine, and that shrank too. So I think they do migrate. At this point, we're not studying migration. And that's the reason also we inject intratumorally, to avoid that possibility that maybe they didn't migrate.
Right.
So, and it still doesn't work, right? So at least we have one less variable. But it's a great question and something that has to be addressed in the future.
Yeah. I had just a question on the CD4 and CD8.
Yes.
So what for these good TAMs versus that, is there a preference for what the CD4 versus the CD8? And can you?
Yeah, so it's a great question. I also asked that question. The preference was for CD8. CD8 only, we saw CD8 mRNA, more CD8 mRNA compared to CD4s.
From the cars?
No, from the total. From the cars, I believe it says also CD8s. But then, we do have more CD8s in the product, so might be that it, but yeah.
You know the percentage?
In the product, it's about 70-30, somewhere in that range.
And do you know how does it change?
How does it change over time? So we never did flow, like taking them out. It's only looking at the single cell, which then I really need to go back and look at the CAR mRNA and then see if the CD4s or CD8s.
Thanks.
So in the HIV field, there are rare individuals, so-called elite controllers, whose immune systems have some properties that let them do something special. So does that ever happen in brain cancer? Are there any elite controllers, and what can be learned about their immune systems?
So there is definitely elite controllers. I know Stanford Group 1 has one complete responder in the APG, so I know they are studying that inside out. In our clinical trial, we have two, also interestingly, the APGs that has stable disease. So we're going to look into that. So, and then again, looking at the APG because it's highly aggressive pediatric brain tumor, there's one patient that got treated with chemo drugs that usually don't ever work. So again, study into that. At this point, again, we're collecting everything we can and waiting for long-term follow-ups to understand what is happening. But I completely agree with you. There is elite controllers. There is those exceptions, but we try to study, understand, is it tumor, is it microenvironment, is it immune response, and all the other, or pretreatment. Unfortunately, what we get, patients that we get for the clinical trials, they went, up to 2 years of different therapies. And so we don't know how that changed and what the response we see. Is it actually CAR T cells or actually their pretreatment?
Okay, we'll take a question from our online audience. CJ Lee asks, do you think it would be possible to reinvigorate TIL by knockout DNMT3A? Can DNMT3A reverse the exhausted epigenetic phenotype?
So I think we can to do that because, because we are reversing epigenetics in the healthy donors, so they are also have particular imprinted epigenetic responses. So I do believe we can. It's just, to expand TILs, it's complicated. Then you do a genetic editing, so you might just kind of end up with a low numbers. So I do believe that you can reverse.
So as I understand, you inject the cells intracranially,
Yes.
let the tumor establish in the brain, and after either 7 days or 25 days post establishment of tumor, you inject the CAR T cells. So do you see a time makes a difference in the efficacy of these CAR T cells in regressing the tumor?
Okay.
Or
Yes, so the 25 was for murine CAR T cells where we wanted to establish tumors and inject CAR T cells to capture, so that we didn't want, if we inject in a smaller tumors, we didn't want tumors to disappear so that we cannot see that population in single cell. So that was another different experiment, but we do have different timings for other tumor models. So I don't have direct evidence if timing matters, but what we do now have for ependymomas, those are very slow growing tumors and we can't predict when we're going to treat them. So we always do MRI. We do MRI and then suddenly once we start seeing tumors, we know that a week later we're going to inject, okay? So, and the data that we collected, we saw, one experiment that worked and the other didn't work. But with MRI, the beauty is that we can actually calculate tumor volume before injection. So we did that and we found direct correlation, which is the moment that there is direct correlation between tumor size and T cell injection. So if it's a big tumor, CAR T cells didn't work. If it's a small tumor, CAR T cells worked. So timing would then, indirect like an answer saying that timing matters. If you have big tumor, it won't work, which is also what we see in the clinical trials because all the patients that come back, they have massive tumors, so disseminated disease, so multiple metastases.
so a follow-up question with that. So if the tumor size is big, will you inject more CAR T cells?
Yeah.
Or
So more CAR T cells don't work. I don't know why. And that's, so yes, but it doesn't work. So I believe, while it makes sense, so I think everyone kind of thinks it's like big, of course it's not going to work. Bigger tumor, less CAR T cells, tumor grows faster and outgrows tumor cells. But I think what's actually happening is just tumor is very different. It secretes more when tumor is bigger, microenvironment is different. It secretes more different suppressive cytokines. And that's why this doesn't work, because when you're right, when we inject more CAR T cells, it doesn't work still. So how come? Like how come it's not working? Even if we make CAR T cells that are better killers, it still doesn't work. So something about that big tumor really not allowing. Yeah. So it might be the other, big study is extracellular matrix. As tumor is growing, do they create some kind of barrier for CAR T cells to get in, or any immune cells for that matter?
So just as a follow-up to the
Mhm.
last point, there are several antibody drug conjugates targeted against the same epitopes that are
Yeah.
pretty effective in the brain. So do you guys think about that combined modality therapy coming in with the debulking ADC and then
Yes.
coming in with the
So
CAR T?
any debulking therapies we're thinking. What worked anecdotally for us in the clinic is that a patient came in, we treated with CAR T cells, an inoperable tumor, and then after CAR T cell treatment it was operable. And then tumors continued, then it was, surgeon did the surgery, then we also were able to treat with CAR T cells and we saw some response. So that clearly combination works and also debulking works, but it's not possible for, like, there's no way you can do debulking for the APGs because of the location, right? So for some tumors, yes, and it can be, and whether it's going to be chemo or ADCs, my lab is not looking at ADCs. I'm actually looking in the chemo drugs. Because I feel like if I find a drug that is FDA approved, I can move, like we can open another arm faster than another modification for time being, and continuously looking at the different modifications.
Okay, we have another question from our online audience. I'm Gasper from Hormel Institute. GL261 cells are highly immunogenic relative to human GBM.
Mhm.
How reliable and translatable the data generated using this model?
This is a great question, great comment, and that's the criticism we received when we were submitting this manuscript, and that's why probably it ended up in very low impact journal, low impact factor journal. However, I think what we found is still like macrophages have particular function, and whatever you're going to do, you're still going to find some patterns. And at the same time, we also see in the clinical trial, we see a response, we see temperature increase, we see patients responding, having pain and all that stuff, but then CAR T cells don't work, as we saw in GL261. So while it's not ideal, it's useful. We learned something, and I'm not saying this is how it's going to be, but for us, it was the first step towards studying microenvironment, and we needed a reliable model. And this is a reliable model where when you inject, after 2 weeks, you're going to have a tumor, after 3 weeks, animal are going to die. When we work with GBM, with DAPGs, we inject in the brainstem, you cannot detect that tumor with MRI, and you're just shooting in the dark. But now that we know what's happening when to study everything, now we're better off. So I started with that. And again, I take all that criticism. I completely agree. It's not ideal, but it's useful. And it helped my lab to establish single cell and spatial work, which was amazing.
Okay, great. We've got one final question online from Zoe Quinn. With repeated infusion of your CAR T cells, did you observe any anti-CAR antibody response?
So we did not have repeat infusions. I'm sorry if I misspoke. We haven't studied yet that. We do repeat stimulation, but we don't have repeat infusions because of we're trying to overcome ICOC requirements because it's just too much. What we did repeated is tumor re-challenge. So I'm not sure if that was the question.
Okay, great. A lot of great questions today. Let's say thank you for a great talk one more time.
Thank you.